Dear all, welcome to our channel. This discussion we are going to continue with in the continuation with our previous lectures where we have discussed about the general structure of the amino acids, their classification. Classification we have discussed the amino acid based classification of the amino acid we have discussed based on the polarity, their metabolic status and the nutrition. In the today's lecture we will discuss about the uh, pep- how the amino acids are linked by a bond that is known as a peptide bond, and the peptide, polypeptide and the proteins, how these amino acids are linked to form a peptide and the polypeptide and the protein. So let's start a lecture. So amino acids, they can act as a acid or base. So if you dissolve amino acid in a water, it can exist in the solution as a dipolar ion, or it is also known as Jutter ion. This term is taken from the German word and which meaning is hybrid ion. So a Jutter ion can act as a acid or a base. So, as we know that acid are having the property of the acids given the solution the protons, whereas the bases they accept the proton. So if you can see that here, uh, amino acid, this is the general structure of the amino acids, and amino acid they are donating here the proton. So they can be known as a proton donor, and they can also accept the proton. So they are working as a proton acceptor. So in this way, the amino acid can also work as a acid by donating the proton, and they can also work as a proton acceptor or the base. So the substance which are having this type of the nature, where with dual nature, they are called as amphoteric substance or the ampholites. So amino acids behave as a ampholites, or they are having a nature of amphoteric substance. This so now amino acids are linked, as we have discussed that amino acids are linked to one another by a bond that is known as a peptide bond. So if we say peptide, so it is a containing a compound which is consisting of two or more amino acid. So if there is a peptide chain of more than 12 amino acids and less than 20, then we'll call it as a oligopeptide. So how this peptide bond is formed? This peptide bond is formed by the removal of a water molecule from the hydroxyl group from the alpha carboxyl group, and the hydrogen from the amino group of another amino acid. So in this way, there is a removal of hydrogen, there is a removal of water molecule, and this is an example of condensation reaction. So the element of the water molecule, the OH and the H, are removed from the uh, two amino acids, and there is a amide linkage is formed, and this is known as a peptide bond. This bond is known as a peptide bond. So the amino acids are linked to one another by a bond that is known as a peptide bond, and this peptide bond is a type of amide linkage. So if there are two amino acids are joined by a peptide bond, if there are three amino acids are joined by having a two peptide bond, and four amino acids are joined by a three peptide bonds, and like in the and so on. So in this way, a long chain of the amino acids can be linked, and a polypeptide is formed. So if there is two terms are used that when here, that is the oligopeptide and the polypeptide. If a polypeptide chains of more than 12 and the less than 20 are formed, then we'll call it as a oligopeptide. If there is a more than 20 amino acids are linked in a chain, then we'll call it as a polypeptide. So there are the two terms, that is polypeptide and the proteins. These are although interchangeable, but and can be used. So here the polypeptide, if but here the terms that are differ based on the molecular weight. If there is a molecular weight is below the 10,000, then we'll commonly call it as a polypeptide. If it is a, although the proteins are having a high molecular weight, but these terms are interchangeable and can be used either polypeptide or the protein. But if we have to consider that if the protein molecule, the molecular weight is more than 10,000, we will generally say it is a protein. So here amino acids unit in a protein, once the amino acid is incorporated in a polypeptide chain or or in a protein, then it is called as a residue. So this is the residue is because here there is a removal of the water molecule. So the left part, which is the left part, that is called as a amino acid residue. So this is one amino acid, like we can see this is the amino acid residue here, and this is the left part that is left for the another amino acid. So in this way the amino acids are linked by one another, one by one. If there is third amino acid will come, then it will also form another peptide bond here, and then it will link. So in this way the amino acids will come and form a peptide bond, and then they lead to the formation of a peptide. And if there is more than, if there is a more than 12 and the 12 less than 20 amino acids are linked, then we will call it as a oligopeptide, and if it is more than 20, then we will call it as a polypeptide. And if the molecular weight is more than 10,000, then we generally call it as a protein. And amino acid unit in uh, in a protein that are commonly called as a residue. So here this picture is showing that this is a polypeptide chain, and there are large number of amino acids are linked to each other. These beads are like a structure. These are the amino acids. They are linked to each other. So there are 100 or more than 100 amino acids are linked, and these amino acids are linked by which bond? That is a bond that is known as a peptide bond. So we can say that protein is a polymer of amino acid. Protein is a polymer of amino acid. So if this here we are seeing the magnified view, that is the, these are four amino acids of phenylalanine, leucine, serine or the cysteine. So this is like a protein or the polypeptide chain. That is polypeptide chain. This is a polypeptide chain where the amino acids are linked by a bond, and that bond is the peptide bond. That bond, that is the peptide bond. So how the polypeptide chain are named? So they are named starting from the amino group. The first amino acid which is incorporated, which is having a free amino group, and there's the conventionally the protein chain are named started from the amino group. So there are two and are free. One is the amino group, and another group you can see find here is a carboxyl group. So these are the so amino group is only free for the N-terminal amino acids, and carboxyl group is only free for the C-terminal amino acids. So there are two terms are we called as N-terminal or the C-terminal. So N-terminal amino acid in the polypeptide chain is the amino acid which is the first starting amino acid, and it is only having the free amino group. It is this polypeptide chain is having the only free amino group, and here the carboxyl group, that is the last other end of the polypeptide chain, this carboxyl group is free here. Other amino and the carboxyl groups are not free, and they are involved in this peptide chain formation. So then we will call it as a N-terminal, and then it is called as a C-terminal, or we will call it as a amino terminal or carboxy amino terminal or carboxy terminal. So here you can say this is the structure of the insulin, and where there are two chains are there, two polypeptide chains, and they are starting the first amino acid here is the glycine, and here last is the asparagine. So this is the amino group, this is the amino terminal, and this is the carboxy terminal here, last amino acid in this chain, and these two chains are linked by a, so here you can see these are cysteine, as we have discussed that there is the cysteine residues are involved into the disulfide bond formation. So this is the amino terminal for this chain, and this is the carboxy terminal. So the one end is amino terminal and another end is carboxy terminal. So like in this way we will say the naming of these polypeptide chains are due according to starting from the amino terminal. So this is the structure of a pentapeptide. So here the amino terminal is here the first amino acid. This carboxyl amino group is free, where the another and that is the carboxyl group is free. So here there are 5 amino acids which are involved. So it is a pentapeptide. There are 5 amino acids are involved and they form a pentapeptide. So first thing is the first amino acid amino group is free, so it is called as amino terminal, and the last amino acid carboxyl end is free, so it is called as carboxy terminal. And we are starting its name, it is like that. So it is a first amino acid residue is serine, second is your, so this is the R chain of the serine, this is the glycine, H group is present, then we will say it is a tyrosine, here there is a tyrosine, so this R group is for the tyrosine, then it is the methyl group which is present here, it is in the alanine, and then it is the isoleucine. So this so its naming, this is the pentapeptide and it's start from serine. So the naming of the polypeptide chain is from N terminal to C terminal. Serine, glycine, tyrosine, alanine and the isoleucine. And so we can say only here the amino group that is free, that is the amino terminal and the carboxy terminal. Other amino groups and carboxyl group are not free here, and they are involved into the peptide bond formation. So the acid, if we will say that acid base behavior of the peptide is produced by its free amino group and the free carboxyl group. So as we have discussed in the previous slide that only the free amino group is present at the N terminal and the free carboxyl group is present on the C terminal at the last amino acid. Other amino acids, amino and the carboxyl groups are involved in the peptide bond formation, so they do not contribute in, so they cannot ionize, so they do not contribute into the acid base properties of the polypeptide. But some amino acids are having their R group which can ionize. So here we can see this is the glutamate which can ionize, so they can contribute, and this is the lysine which is having extra amino group. So these R group can ionize and they can contribute into the acid base behavior of a polypeptide chain. So the number of the amino acids and the nature of the amino acids, which type of the amino acids are present, the amino acids which are having a polar positive charge or the negative charge amino acids, the what are the number of the positive charge amino acids and what are the number of the negative charge amino acids. Based on this and their nature and the number of the amino acids determine their ionization behavior, because all these amino groups and the carboxyl group, they are not free, and only the amino group terminal and the carboxy terminal groups are free, but the R group which are present for some amino acids, so that contribute toward the acid base behavior, and their number will determine the acid base property of that peptide. If there is a large number of glutamic acid or the aspartic acids are present, so they will more having a negative charge amino acids, and they will have more the acidic properties. And if there is a more basic amino acids are present, so this type of the peptide will have a basic type of the properties. So now comes to the biologically active peptide and the polypeptide. We have seen that these amino acids are linked and they can form a biologically active peptide and the polypeptide. So you can see that the very small number, like if there is a peptide of 3 to 4 amino acids or 10 amino acids, they can also have a very significant role in the biological system. And there can be a amino acids which just the peptide which are of 3 amino acids, they are also having a very significant role. So that we will discuss about some of these peptides which are like 3 to 10 amino acids or 20 amino acids, or like in the range of 30 amino acids, and they are having a very significant role. So the example you can see here is a dipeptide, that is a aspartame, which is the L-aspartyl and the L-phenylalanine. It is a dipeptide which is made up of two amino acids where the aspartic acid and the phenylalanine are linked together, and it is known as a aspartame, or it is a artificial sweetener. It is known as a artificial sweetener. It is not a sugar in nature, but it behave as a, its taste is very sweet in nature. So it is a dipeptide and is tasted sweet. So it can be used in a as an artificial sweetener. And various whatever hormones are also there, which are small peptide. Example you can see here is the oxytocin. Oxytocin is just a 9 amino acid residue and it is secreted by the posterior pituitary. So you can see it is a very small peptide, that is oxytocin, just 9 amino acid residue, and it is having a very important function. It is having a role during the uterine contraction. So it is secreted by the posterior pituitary and stimulate the uterine contraction, help into the parturition. Another is the bradykinin, that is just the 9 amino acid, and it inhibits the inflammation of the tissues. Another peptide, that is the thyrotropin releasing factor, that is having just the 3 residues, and it is formed in the hypothalamus and stimulate the thyrotropin from the anterior pituitary. It is stimulate the production of the another hormone, that is the thyrotropin, and from the anterior pituitary gland. So it is clear that these are peptides which are very small in size. They are also having a very important role, and they can act in a very small amount, such as hormones such as oxytocin, thyrotropin releasing factor, which act in a very small amount, and these are very small in size. But so they can also work as a and so they size although is small, but they can have a very significant role. There are some proteins are also there, or the polypeptide, which are very large in number, large in size, and they are also having a very important role. So the biological active peptide, polypeptide, they can have in a very vast range of the size. Another example is the insulin, the glucagon. There is a pancreatic hormone insulin, which is secreted from the pancreas beta cell, and it is containing the two polypeptide chain, and one is containing the 30 amino acid residue and another is containing the 21 amino acid, and this insulin is having a very important role in the carbohydrate metabolism in the body, and its deficiency or its not proper functioning can lead to the diabetes. So here, so peptide insulin is also a polypeptide. It is made up of two polypeptide chain, that is one is having 30 amino acid residue and another is having the 21 amino acid. So another hormone, that is the glucagon, which is also a polypeptide in nature, that is the 29 amino acid residue, and it opposes the action of insulin. So these two hormones which are polypeptide in nature and are having a very important role. So we can say that the amino acids can link on a diverse size, and they can have a very important role in our body. So this table is showing the molecular data of some protein, that is like cytochrome C, ribonuclease A, and these are, you can see here, this is having a 104 amino acid residue. It is made up of 124 amino acid residue, and here the polypeptide chain is just a one polypeptide chain. That myoglobin, which is also a very important role in oxygen transport, and it is having a number of residue is 153, and it is made up of one polypeptide chain. Another example you can see here is the chymotrypsin, which is the having a polypeptide chain R3. So there are three polypeptide chains are there, and its number of residue is 241, and its molecular weight is 21,600. So this is the list of the some proteins which are having a molecular weight more than the 10,000, and number of residues can also vary. So this table is showing that the proteins which are found are having a diverse type of the function in the biological system, and they are having a diverse size. Some can be 100 amino acid residue. Another protein that is here is a titin, which is around a 26,926 amino acid residue. It is the one of the largest protein, and these amino acids are linked in a single polypeptide chain, and its molecular weight is around very high. So these protein, you can see that the proteins, these are having a diversity in their size. They can be of very small in size, like 3 amino acid, that is peptide, that is TSH, that oxytocin that we have seen is the 9 amino acid that can have important role, and the or they can vary. There, like hemoglobin, there is 574 amino acid residues are there, so they are also having a very important role. So the size of the peptide, polypeptide, and the amino proteins vary in a vary, and their function is also vary. So in the previous slide we have seen that the some proteins they are having a one amino polypeptide chain, some are having a multi subunit protein where 3 or 4 protein polypeptide chains are linked together. So in this we will discuss about the multi subunit proteins. So some protein that we have seen that they are containing only a single polypeptide chain, but there are some protein which are made up of more than one polypeptide chain. So they are containing either 2 or more polypeptide chain, and if they are associated non-covalently, then we'll call it as a multi-subunit protein. So the multi-subunit protein, there should be a more than one polypeptide chain. So there can be 2 or more polypeptide chain, and they should be associated non-covalently, and such type of the proteins are called as multi-subunit protein. So these individual polypeptide chain in a multi-subunit protein can be identical or different. So that if there are two subunit in a protein, so these two subunit can be identical or they can be different. So another term is used here, that is the oligomeric protein. If the subunit, if a protein that is containing a 2 polypeptide chain, and if they are identical, then we will call it as a oligomeric protein. Then we will call it as a oligomeric protein. And the identical units which are present, that are called as protomers. This will be more clear with the help of an example. That is the example we have taken with the hemoglobin. You can see hemoglobin, it is a made up of 4 polypeptide chain, and these are 2 identical alpha chain and the 2 identical beta chain. You can see here, these are the 2 alpha chain and these are the 2 beta chain. These two are both identical. So or-fold so it is a, we can say it is a multi-subunit protein because it is containing more than one. There are 4 polypeptide chain, and these 4 polypeptide chains are held together, associated with the help of non-covalent interaction. Here you can see that each alpha subunit is paired in an identical way with the beta chain. So we can say it is a, so here the identical unit which is that is the alpha beta subunit. So this alpha subunit is paired with a identical way to each other. Like alpha beta, these are here the same identical binding takes place, which is here with the alpha and beta chain. So we can say it is a hemoglobin is a tetramer of the 4 polypeptide chain, or it is a dimer of the alpha beta protomer. So here the identical unit is alpha beta. So it is the alpha beta protomer, and it is called as a dimer of the alpha beta protomer. So So it is a multi-subunit protein. So here the 4 chains, it is a tetrameric protein here, and this protein is held together by these, all the 4 chains are bind together with a non-covalent interaction. Here the difference comes if the individual polypeptide chains are called a chain. So this is the structure of the insulin, which is having a made up of 2 chain, A chain and B chain. Here we will not call it as a protomer because these 2 chains are not identical to each other, and these polypeptide chains are linked covalently. Here these chains are not associated non-covalently. There is a covalent bond formation, there is a disulfide bond formation takes place between two cysteine residues. So these 2 chains are linked with a disulfide bond. So if these the 2 polypeptide chains are linked covalently, then we'll call it as a chain, not as a protomer. So how we can, now we have discussed that the amino acids, they are called as a residue. The term which we use as a residue after the removal of the water molecule, because this water molecule is removed during a peptide bond formation. So how we can calculate the approx number of the amino acid residue in a simple protein? Simple protein, we are not saying that there is some chemical modifications to that any protein. So if there is simple protein, so how we can calculate the approx number of the amino acid residue? So there is, like, we can see that the average molecular weight, it is find that if you take the average molecular weight of the all the 20 amino acid, it is around 138 amino acid. But it is found that the small amino acids are predominant specifically in the most protein. So if we consider it, then you will find that the average molecular weight comes with the 128 because small amino acids are predominant. And as we have discussed that during a peptide bond formation, there is a removal of water molecule. So average molecular weight is 128. So there is a removal of water molecule. So we can minus it. So 128 minus 18. So it will be 110. So this is the, like, we can, if we are having a molecular weight of a protein, we can divide it by the average molecular weight of the amino acid residue, and based on this, if we are having a molecular weight of a protein, divide it with the average molecular weight of amino acid residue, and then we can get the number of amino acid residue. Like if we are having a molecular weight of a protein that is 4400, we know that the average molecular weight of amino acid residue is 110. So if we will divide it, then we can say this protein is having a 40 amino acid residue. In this way, we can calculate the approx number of the amino acid residue in a simple protein. Next point is that amino acid composition of the two protein. This, like, you will find that this table is representing the data of the presence of the amino acid, different type of the amino acids. This one protein is bovine cytochrome C and another is bovine chymotrypsin G, chymotrypsinogen. So you can see here the alanine, the number of the amino acid, that is alanine, is 6 here, and here is 22. You can see, if we will take another, that is asparagine. Here the number of the asparagine residue is 5 and here is 15. So it is not that all the 20 amino acids will find in the equal number in all the protein. So these amino acids, the number will vary. All the, like, if alanine in a one protein it is found the 6 in number, another protein it is found the 22. In third protein it can be in the number can be 18. So the frequency of the amino acid can vary here. You can see the glycine here is the 14. Here the amino acid residue of the glycine is 14 in the this protein, and here is a 23. So some amino acid can occur once, or some you can find the presence of some amino acid only once, like serine. Here it is just one, whereas the in the bovine chymotrypsin it is 28. So the number will vary very, like there is a huge variation into the number of amino acids. So this is not that equal the number amino acids are found in equal number. So it is like one amino acid and it is a very large number, that is 28. So this makes that the proteins are having a different function, and this is because the presence of these amino acids. The relevant, like the number of the amino acids, which type of the amino acids are present in the protein, and that makes the different function of the protein. So these two proteins differ in function, and they also differ into the relative number of the amino acids. So based on the similar thing, if we will compare the 4 or 5 amino proteins and then see the amino acid composition, so you will find there is a huge variation between one protein to another protein. Next topic is that you will find that some protein, they are containing some permanent chemical group other than their amino acids. Like some proteins, you will find there is a, apart from the amino acid, there is some chemical group is also present, and it is present permanently to this protein. And based on this, these proteins are named. So this chemical group, that is the permanent chemical group that is attached to a protein, it is called as a prosthetic group. So it is the non-amino acid part of the protein. It is a non-amino acid part of the protein. So like there is a protein, it can have a two part. One is the amino acid part, and it is a non-amino acid part. So the amino acid part of this protein is, that is protein in nature, and non-amino acid part, that is, we'll call it as a prosthetic group. So the protein which are containing this non-amino acid part of the prosthetic group, such proteins are called as a conjugated protein. Such proteins are called as a conjugated protein. And in proteins in which there is no prosthetic groups are present, such proteins are called as simple protein. There is no chemical group is in the protein. They are only made up of simple amino acids. So such proteins are called as simple amino acid. And in some protein, the chemical group is attached permanently. So such proteins are called as conjugated proteins. So the example you can see here, it is a lipoprotein. So it is containing a lipid. So it is called as lipoprotein. Example is beta lipo, one lipoprotein of blood. Another protein, that is a glycoprotein. So it is containing some carbohydrate as a chemical group. And the best example is immunoglobulin G. Some protein are containing the phosphate group. So it is called as a phosphoprotein. And the example is casein of milk. Hemoprotein, they are containing heme as a chemical group. It is permanently associated. All these groups are associated permanently. So it is a hemoglobin. Flavoprotein, it is a containing the flavin nucleotide, and the example is succinate dehydrogenase. Some protein containing some specific metal such as iron, which is present in ferritin, that is zinc, that is present in alcohol dehydrogenase, calcium, that is present in calmodulin protein, molybdenum, that is present in dinitrogenase enzyme, and copper, that is present in plastocyanin. So you can see here that some proteins containing some permanent chemical group, and these proteins are named as conjugated protein. So that now we have discussed about the amino acid, their classifications, and how the amino acids are linked to the form of protein. So that is by the peptide bond. Some common term associated with these proteins, that is the polypeptide, how the polypeptide chain is formed, how it is differ from peptide to polypeptide in the protein. So that we have discussed that it is based on the difference into their molecular weight. If it is more than 10,000, then we'll say it is a protein. If it is less than 10,000, then we'll generally say it is a polypeptide, but these terms are generally interchangeable and can use uh, interchangeably. Then we have discussed about then what is multi-subunit protein, how we can say by the chain in a protein. So like these terms which are used generally in a protein, so this common term that we have discussed, the conjugated protein, simple protein, how we can calculate the amino acid number uh, in a protein, so these things we have discussed. In the coming lecture we will discuss about the how we can purify the protein from a cell or a tissue. If we are having a protein, and how we have to identify its characteristics, then how we can identify this protein. So this in the coming topics we will discuss about the different techniques which are utilized to isolate a protein and the purify a protein. So these topics we will discuss into the coming lecture. So hope you have like this series, this lecture. So if you like this, please subscribe and like our channel. Thank you.